Well, I thought it would be a great idea if we could get together and ask the most pertinent, important, salient questions in the field of nanofossils, coccolithophores. There's a huge number of people working on the field. As you can see, I showed this at the INA in Santos. The number of publications of coccolithophores per year is going up, skyrocketing. And because there are a lot of questions regarding coccolithophores that have been unanswered. And there are lots of facets to coccolithophore studies from, biology, geology, I'll mention them soon. And I don't think I'm the one to, absolutely know which these important questions are. It should be a community effort. And I think it would be a great idea. Maybe we could put that up on the INA webpage so that we could, better focus our work. I think everything is important, but I always feel asking the important questions helps me focus better, and perhaps will help others in the field. Now, that's the nice thing about coccolithophores. There's so much to it, to studying them. And of course, they've been my favorite organisms. When I started in the 70s, sorry. In the 70s, there weren't very many workers in the field, and now it's just blossomed, which is great to see. And the driving questions, I just put up here all the different fields, and they all interrelate with each other. There's no linearity between one and the other. So they all interact. So we have biogeography, biochemistry, biogeochemistry, biostratigraphy, quantum biology, a brand new field, which is really interesting. And I suggest you look that up in Google if you can. There's some fantastic things. I can't get into it. Molecular biology, sedimentology, satellite biology. There's so much that my human brain can't fathom it. So eventually, we should be getting into the modeling aspect and trying to understand it that way, perhaps. And so, for you who have questions that I haven't put up, I couldn't put everything up. I put it in the chat, or send it to Felipe or INA. I think it would be, or me, and we could put it together in the INA webpage, Jeremy permitting. So we could ask. We can put it in different questions. It's always hard to make a diagram to interrelate everything and put it in a succinct way. That's the trouble, when you write, you don't know which comes ahead of the other. The order is important. Unfortunately, we have only 3D minds when, this is actually 4D dimensions. But here are some of the questions. One of the most important, I think, is what would the world be like without coccolithophores? I say this because I have talked to many people at NASA and and modelers and other people who've really never heard of coccolithophores, and we have to spread the gospel. If you talk to the average person, also I digress a little bit. If you talk to the average person on the street and you ask them what is a coccolithophore, they say, "What are you talking about?" And then I give them my 10-minute talk. And I have another person who knows about these important organisms. I think we could do a much better job at telling other people around the world, politicians, whatever, help the funding, if people knew how really important they are. But some of the people I've talked to, and some of the proposals I've written say, they've answered the reviewers here as references. What other organisms would take up the slack? Would it make a real difference? We don't think so. Proposal declined. And so our community needs to really think about how to answer these people. I know we always have the same paragraph, coccolithophores are important in the biogeochemical cycle and so forth and so on, but I think we've got to do better than that, and it would be great to become more succinct and focused. So then the next question I put up here is what makes them so successful? Is it because they produce coccoliths? The coccolith factory, I mean, they spend a huge amount of effort, we saw from Jeremy's talk, producing these things, and we really still don't know what is going on there. Would be great to understand that, and it's been going on for a long time. So they must have a very good reason, but they don't talk to us in our language, so we have to figure it out. And then there are other questions, what percentage of the oxygen we breathe comes from them? I really have never understood. I don't know what percentage it is. I mean, Balch and many others, and I've read this in textbooks, 50% of the world's ocean. But those include the 3 major marine phytoplankton. So what is the actual role of Coccolithophores? What percentage of the carbon pool do they represent? Well, 1 to 20 is a large spread. Is that true? Of the phytoplankton carbon pool? I have some references here. And I say this is not these questions are not exclusive. There are many, many more that could go in here and could be organized in a much better way. I'm not very good at organizing things. What percentage of the ocean sediment? Well, not many people have worked on this recently. Brooker and Clark, I spelled Brooker wrong. 30 to 90% of the carbonates in sediments. What is their role in the biogeochemical cycle? Okay, that's a big one. I just put in one little thing here. Contribute up to about half a petagram of calcium carbonate. One of my pet things since I've started is, do Coccolithophores and diatoms communicate with each other? So I think we shouldn't look at Coccolithophores only by themselves. We need to integrate the entire community in the oceans and try to understand it. Yeah, I mean, Coccolithophores, I'll go into this soon. Okay, so what is the role of the deep photic layer, which has been consuming me lately? Then we have, where are you here? Actually, who eats them? It's been said copepods. What is their real role in the food chain? Maybe I haven't done a wide enough literature survey, but I don't think we really understand everything that's going on there. Sinking rates are important. I just read a nice paper by Sarah Milner. How they affected by temperature. I think it's 2014, not 2004. Do they increase or decrease calcification in a more acidic environment? Well, there's going to be winners and losers. Do we really know what's going on? Oh, there's so much work to be done. How exactly are the holococcoliths produced? There are many questions that have come up during these talks I just put into, and the reliability of biohorizons. I would love to get together and organize these questions. And by the way, asking the questions helps you think through the processes involved. One of the things I've always been, why have Braurodosphaera and Miliana been so successful? And so I'm thinking they have, this is not the only thing that makes them successful. This is just one of the ideas I've had. Well, they both have naked haploid phases, and they're both mixotrophic. So Kamenaya et al. In 2018 showed that they are pomocystoses. They can eat organic matter, and Emiliania, there's a paper that came out this year showing that they do the same. So are all Coccolithophores mixotrophic? Are they only the ones in the deep photic layer, which really have to be in order to survive down there? So these are really fun questions to ask, I think. I'm trying to figure out what the time is here. Okay. How are Coccolithophores responding to global warming? I put this chart together a couple of years ago, showing that they are moving, because we have 30 years of satellite data right now. Unfortunately, not the same sensors all the time. There was color zone, color scanner, CWIFs, MODIS. Putting them all together has been a big chore. And so in these diagrams, you can clearly see that they're moving northwards and southwards. And I read the paper this morning in the New York Times, actually. A science article just came out showing that the Arctic regime has shifted entirely. They did new primary production, NPP. They have a really nice algorithm. It showed that the entire NPP change is changing in the Arctic region. Soon they'll find out the same will be in the Antarctic, around the Antarctic. The Arctic is really a fascinating thing. I have a student, Haiping Jiao, who just produced this at 4.6 kilometer resolution. It's just amazing. And what's happening now, which was and is happening more and more, is that the Coccolithophores, and we look at PIC as a proxy for the calcifiers, are producing double blooms. So in May and August, before it was only a single bloom, which you can read in all the books, but now they're starting to produce double blooms, and that goes very well with the new Science article. So things are going on right before our eyes, and so we as a community, could be spearheading a lot of what's going on. So this is continuing. We're doing this for every year when the satellite was up. And we're going to try to relate this to changes in the wind direction, sea surface temperatures, and other important variables. So this will be a fun project. So the role in the biogeochemical cycle is probably one of the most difficult ones, because if you try to put this together, I showed this at the INA meeting in Santos, there's a lot of things that go on here. And so you really need modeling. Of course, you can say that it reduces DIC, dissolved inorganic carbon, by 1 and total alkalinity by 2. We know it drives down alkalinity. Not really a good thing in the short term. This is psi here. This psi here is the amount of CO2 produced versus calcium carbonate, and a ratio, and it seems to be 0.66. This number is quite important to understand the PIC-POC ratio and all the other variables. So we really need to get a very good handle of this in the models and to be able to reconstruct it. And so there's also the ballasting and all kinds of other things that are happening. One thing would be great to have a better handle on the haploid Coccolithophore life cycles and understanding how they affect the biogeochemical cycles. There are at least 100 questions just from this. And of course, mixotrophy, which is really important, because if they're mixotrophic, of course, the biogeochemistry is going to change a huge amount. And so once they go down on the ocean floor, we're completely in a different time range here. So no longer in the 10-year time range, we're into the million-year time range here. And so that combining the different time ranges together in modeling is extremely complicated. But we know the classical view of this, but really very good understanding is still far away. And of course, they essentially pump bicarbonate alkalinity from the surface to benthic waters. And then once they're gone, that's good. But right now, producing more CO2 than depositing calcium carbonate is a problem. Now, diatoms are in there too. And diatoms are doing something completely different. Diatoms probably are better for the short term than Coccolithophores, because they're producing CO2 in this process. And again, there's so much work that needs to be done, but we need to go out in the field. And there have been really very few oceanographical trips that go through the entire year. We need one or two ships dedicated for to be out there sampling the water for 2 years. Any volunteers? Let me know. We'll write a great proposal, all of us, and we'll do it. We'll go on the ship and we'll talk Coccolithophores all the time. Well, nearly all the time. Okay. So here's this reticular fenestra sessilis. Yeah, I discovered. No, I didn't. I saw it in the Red Sea even in 1970s, and you can see these diatoms are always around. The Coccolithophores are always around the same diatom or nearly similar diatom species. There's something going on here, and therefore I think they must know what they're doing, and they must communicate with biochemicals with each other. How about growing them in culture together and seeing what happens, and then spray in a little bit of dinoflagellates while you're in it, doing it. So now one of my pet things is deep photic layers, and so my apologies for those who have already seen this, but you have the chance to look at it again. So my thing is, since the beginning, these satellite photos are great, showing you the chlorophyll distribution, but it's only from the surface. And you can see that these organisms, well, they say satellites are good to 70 centimeters. But Coccolithophores don't really love it up there, photo inhibition and all that kind of stuff. So they actually like to live further down. And these blue areas here are so-called oligotrophic. But I've been saying all along that Coccolithophores are living down to depths of 400 meters in certain places, and even though they might be much less abundant, the volume of this area, because it goes down to 400 meters here, in these areas there's photosynthesis down to 10, 20 meters. If you could do all the volume, it equals or best the volume of these productive areas, and really we need to look at these much more closely. There's been very few studies made of them. I don't have really time, but this is a model of these. Well, I'll go through it quickly for you guys. Here you can see, this is a great model of the primary productivity from the surface. And it's extremely accurate, but Titania, who's doing this, really hasn't heard much about Coccolithophores. She would love to collaborate with any one of you to figure out what's going on deeper in the water column. You can see how beautiful this is. So let's do that. Let's get together with modelers. We have to talk to them. And modelers are still not there in terms of what we know about Coccolithophores. How do I stop this? Okay. So this is a Pisces model showing you with latitude. You see this is what we saw in the satellite photo. They like higher latitudes, but look at this. Here they are down here in the lower photic zone and below the lower photic zone, living happily thereafter. And again, you can see over here, all of these are living down 150 meters. Here's the usual profile in the deep, in the gyres, and down to about 350 meters. These are Coccolithophores and other organisms. You can see here that this is the estimated chlorophyll, A, at the DCM. So this is sort of the inverse of what we see from satellites. You can see especially over here in the tropical Atlantic. But even here, where has the lowest, so-called lowest productivity, you can see Coccolithophores. Here's a diagram from Florispera profunda by Cortez, and you can see this is sort of a 2-year diagram, and you can see that they do live down there and are very important, considering that the skeleton produces much more calcium carbonate than Emiliani Huxley does. Also, I'd like to draw your attention to Luc Beaufort's work that he has. He was down there in the South Pacific, in the oligotrophic South Pacific, the most extremely oligotrophic area in the world, and he discovered many deep photic layer Coccolithophores. So here's the deep chlorophyll maximum, and Below in the deep chlorophyll maximum, we don't have that many. We have different Coccolithophores than in the deep photic layer. This is called the shade community over here. And you can see that in the shade community, you have abundant Coccolithophores over there. Unfortunately, we can't, well, Ian Probert and Kyoko and others are trying to grow them. Eventually, I think we'll get there. We'll pick up one of these guys and we'll be able to grow them. We'll know more about their genetical peculiarities. My student, Josue, who's listening now, is going to be working on that too. So, you know, we have to understand. Now, they have to be mixotrophic, really, because there's just not enough food down there. Oligotrophic region, you know, so they take advantage of the little sunlight they get, and they're also above the nutricline, where they can get a little nourishment, but certainly not enough. So I think these, we'll find out that they're all mixotrophic. Of course, Amelia neohuxlei might be a little different down there than in the surface, so that's another great study to be made. Okay, and down there we see that they have actually enhanced calcification, increased production of pigments, size reduction. I'd love to study them from our 2-year ship cruise. Hey, there's enough money out there for us to really get a really lovely international proposal together to do it. So this is kind of the modeling that needs to be done. And there's a lot of stuff that is involved in trying to figure out the biogeochemical cycle. And here I have diatom species together with Coccolithophores, the other organisms, the pick-pock ratio. Oh, there's so much stuff. And also the physical chemical parameters I need to go into here. We're just at the scratching the surface, let alone the deep photic zone, to understand what's happening. This is another model. This is the first one, Ernst Mayr Reimer. He was the first one who thought about these things, and without him, we wouldn't be anywhere. And all of the models go back to this one person, and you can see all the things that go into these kinds of models. But none of them have Coccolithophores as a functional group. So that's what I'm doing now. I'm working with Manfredi at Scripps, Manfredi Manassi. I'm trying to convince him to put in Coccolithophores, and he said he can do that. So I hope we'll see what's happening. His program is called well, Pisces, and he's got one called Darwin, which is a program that, again, tries to coordinate all the different variables. But what's missing here are the observations. And so, well, you can have great models, but we need way more observations. So there's a really exciting time to be around and to work on Coccolithophores, and I hope I've given you motivation to continue your studies and to come up with the big questions, to talk to one another, find out what's going on. It's really hard to keep up with the literature, but I think we can do it. And so the 2 suggestions I've come up with is, let's write the important salient questions, and also we can work on a proposal to go on a large research cruise and do all of us working on that to get some of the answers and put Coccolithophores on the map. So 10:30 exactly, and so I thank you, and here is wishing you all the best in these COVID days from Rincon, Puerto Rico.